Hi, my name is Michael Kramer, and I'm going to discuss topics in fluorescence microscopy. Fluorescence microscopy is a technique used to visualize autofluorescent cellular structures, fluorescent dyes, or to analyze the expression or modification of proteins using a technique called immunofluorescence. In order to properly incorporate fluorescence microscopy into an experimental design, one must first understand a few simple but crucial concepts. When comparing fluorescence microscopy to a more traditional method of microscopy, such as bright field microscopy, there are a few key differences that should be noted. The first of which is the light source that's used. So let's take a look at bright field first. Bright field microscopy takes advantage of incandescent light sources or white light sources. So your source contains all wavelengths of visible light. So you're exposing the specimen to many wavelengths of visible light. Whereas fluorescence microscopes require the use of a near monochromatic, and this light source usually comes from an excitation laser or a mercury vapor lamp. If you're using a mercury vapor lamp, you need to couple that with the use of filter sets in order to filter out any unwanted wavelengths of light. Next, we should take into consideration the way that these light sources are utilized in each type of microscopy. So here's your light source, and it's passing through what's known as a condenser lens. And that condenser lens is going to condense the light, shine it through the specimen, and then it's going to pass through the objective. And after it passes through the objective, the light is further condensed through the ocular lens, and that image is portrayed. That image portrayed is what the viewer sees. So now moving on to the way this light is utilized in fluorescence microscopy, I'm going to assume that the light source is a mercury vapor lamp, just so I can show how these filter sets are used. So this mercury vapor lamp is going to shine, let's just say 3 different wavelengths of light onto this first filter. And this first filter is called the excitation filter. And each fluorophore has an optimal excitation wavelength. So let's say that the fluorophore on this sample excited at 550 nanometers, which is going to be green light. So this excitation filter will allow the 550 nanometer light to pass through and reach the specimen. So when it hits the specimen, it's exciting the fluorophores, and then it's emitting light at or lower frequency, so there's less energy in the light. So it's going to pass through the dichroic mirror, and then it will also pass through the emission filter. And then that is the image that the viewer is observing. So a fluorophore is a fluorescent chemical compound that can emit light upon excitation with light. When we excite a fluorophore with a certain wavelength of light, that it's going to emit light at a longer wavelength. So we're exciting with this wavelength, and the fluorophore is emitting with a longer wavelength of light. So let's kind of take a look into what happens on the atomic level. So when we're thinking about this concept, there are 3 energy states that we should be concerned with. The first of which is the ground state. And the ground state is the state at which the electrons of the fluorophore are at their happiest, most content state. So the next is a more unstable, excited state. So when we excite this fluorophore with this wavelength of light, we're actually exciting the electrons and elevating them to a higher and more unstable energy level. So from there, some of this energy is lost through molecular vibration. And when this energy is lost through molecular vibrations, it's converted to a heat form of energy. So that energy is lost, and now these electrons are in a more stable but not very stable state. So that's going to be called a semi-stable excited state. And eventually, these electrons will return to their original ground state. And then this right here, the energy emitted as light, is what we see when observing our sample with fluorescence microscopy. So as a result of this energy loss, energy is emitted in the form of visible light. So we're emitting at this wavelength. So this process occurs very rapidly. It's actually once every femtosecond around, so that's 10 to the minus 15. So it's pretty incredible how fast this process is really happening. And because our eyes can't really detect the rapid cycles of excitation and emission, we see this as one continuous emission of this red light. So because there's energy lost throughout this process, we can graphically depict the expected excitation and emission behavior of a given fluorophore of interest. So this can be done by plotting frequency as a function of wavelength. So just as an example, let's take a look at Evans blue dye. This is a very commonly used dye. It has a very high affinity for plasma albumin. So Evans blue dye is excited at a few different wavelengths, but its optimal excitation wavelength is going to be 620. So when I say it can be excited at other wavelengths, take into account these other wavelengths that can also excite it. But this is where we're going to get the most emission when we excite at this wavelength of 620. So then the emitted light is going to be around 680 nanometers. So this is the optimal emitted light peak. And as I stated before, that energy that is lost is accounting for that change in wavelength. And that's what's known as a Stokes shift. And a Stokes shift is just the difference between the excitation and emission maxima values. So it would be pretty inefficient to try to excite a fluorophore when we know its optimal excitation wavelength. It would be pretty inefficient to try to excite that fluorophore at let's say we're looking at Evans blue. It would be inefficient to try to excite it at a wavelength other than 620, because when we're exciting at 620, we're getting an optimal emission at 680 nanometers. So let's just take a look at maybe some differences that would occur if we excited with a different wavelength. So we'll stick with our natural 620 peak, and we'll show here that optimal peak at 680. This is our emission, this is our excitation, and we'll give this peak an A. So we're associating this excitation at 620 with this emission at 680 at those optimal amounts. So now let's say we excite around 610. You take a look at the emission peak that's going to be able to be detected, and you're going to get a decrease in the amount of light that's emitted and detected by either your eye or the camera that you're using to examine your specimen.